Hello my dear students, welcome to our channel Rajesh Biology and I am Rajesh MSc Zoology. Today our topic is gastrulation. That means before that also we are going to discuss about the bilaminar embryonic disc. How the bilaminar embryonic disc convert into the trilaminar embryonic disc by the process of gastrulation. This is I am going to discuss in this chapter, in this class. Okay, not only the gastrulation, neurulation also. So first we will start from the implantation process. Here if you observe, the implantation process we already completed in the previous class. Okay, that implantation starts after the 6th day of fertilization. Once the fertilization is completed in the January 1st, for example, fertilization is completed. January 6th or January 7th, the implantation process will be begin. Okay, and by sending that syncytial trophoblast into the endometrium and by getting the nutrients, and it develops trophoblastic villi. Okay, cytotrophoblast, syncytial trophoblast, completely embedded within the uterine wall. It takes 1 week time. That means at the end of the second week, the implantation process will be completed. The process of implantation will be completed at the second week after the fertilization. That means nearly 13th day, 14th, January 13th, January 14th, if fertilization takes place at the 1st January. Okay, come back. So in previous video, just did a small mistake. That is what the inner cell mass is towards the uterine wall. Where there is an inner cell mass, on that side only implantation takes place. On other side there is a blastocele. Okay, this is implantation. Now in this video I am going to explain what will happen to the blastocyst after implantation. What will happen to that embryo? This is called embryo up to the 8th week of development. Still it is 2nd week only, right? Okay, up to the 8th week we call it as a embryo. From the 9th week only we call it as a fetus. So in this embryonic development, what will happen? This is outermost, this green color lining is syncytial trophoblast, and the inner what? Cytotrophoblast. Okay, and this blue color, this is embryonic proper, which has the stem cells. Stem cells means which can develop into pluripotent stem cells, which can develop into the endoderm, ectoderm, mesoderm also. Okay, all the stem cells undifferentiated, still it is not differentiated. Okay, undifferentiated stem cells are present here. This is embryonic proper. And this cavity is called blastocele, in which that it is also considered as the primary body cavity. Okay, this is the situation, and it is completely implanted. It is completely embedded. It is embedded within the uterine wall. Then I am taking this only. That means only the trophoblast and inside the inner cell mass. From now onwards I am explaining this. Okay, bilaminar embryonic disc. How the bilaminar embryonic disc is formed. Okay, the cells first differentiated into the two layers. This is the first differentiation. Okay, up to now all cells are equal, but later they are adjusted. They are adjusted in two layers like this. One is blue color layer, another one is purple color layer. I draw here. Okay, so like these two layers they adjusted. One layer which is outer side, that layer is called epiblast. That is called what? Epiblast. And another layer of cells which faces the cavity, which faces the blastocele, that is called hypoblast. Epiblast is the layer of cells which faces the villi, which faces the syncytial trophoblast, that is called epiblast, outside. Inside, which covers, which lines the blastocele, which faces the blastocele, that is called what? Hypoblast. Okay, that is the first thing. Like this, first differentiation of cells takes place. After this, what happened? This hypoblast lines the cavity and it get delamination. Delamination means what? You know lamination process? Lamination process, for example, if you take your Aadhaar card to any Xerox center or some lamination centers, they will keep two covers. In between two covers, they keep one Aadhaar card, and in that lamination method, both are attached with one another. They both come closer. Okay, this is called lamination. Delamination means already attached layers get separated. This separation is called delamination. How this delamination process takes place? For example, if it is the trophoblast. In this trophoblast, the cells embryonic proper is actually attached like this. Okay. After delamination process, what will happen to this? In the delamination process, they will move away from the trophoblast layer. They come into the middle like this. What happened? Already attached cells get delaminated. They are separated. They comes down. Okay, like this, they are delaminated, and the blastocele cavity is spread throughout this embryo. Within this, it will spread like this. Okay. Okay, within this also delamination process takes place like that. One cavity is formed. Okay, now this stage is called bilaminar embryonic disc. Why it is called bilaminar? Because here there is two layers. Before differentiation, there is only one embryonic cell proper, okay, or inner cell mass. There is no cell differentiation. Once the cells get differentiated into the epiblast and hypoblast, two layers of cells is there. That's why it is called bilaminar. Still it is before the 9th week, so it is the embryo, bilaminar embryonic, and it is in the disc shape. You know, DVD, CD, how they look like. As like that, the cells are arranged in this manner. Okay, and it is covered by the trophoblast. Okay, it is attached to the trophoblast. You can see like this. For example, if it is the trophoblast, this red color lining, what it is? Trophoblast. Okay, with the trophoblast also there is some cells is there, and these cells are attached like this in this two-layer manner. So bilaminar embryonic disc shape. So to show that disc, just try to draw a 3D diagram in this manner. You can observe this middle part. This middle is the disc-like structure, and that is the covering. So it is called bilaminar embryonic disc. Still the gastrulation process is not yet started. Okay, before it starts, you have to know what is gastrulation means. Now, gastrulation is a dynamic process. Gastrulation is what? Dynamic process. Dynamics means movement, which is in the locomotion, which is in the movement. That is only we call as a dynamics. Okay, so gastrulation is a dynamic process, means cells are moving. Okay, so when the cells are moving, the gastrulation process will be start. Because of the movement of the cells, because of the gastrulation of the cells, bilaminar embryonic disc convert into the trilaminar embryonic disc. So gastrulation is a simple migration of cells, movement of cells is called gastrulation. How the gastrulation takes place? In this embryonic disc, I am taking this embryonic disc out, I am keeping here. This is the embryonic disc. Okay, so in this embryonic disc, in a longitudinal axis, one streak is formed. One streak. Okay, the primitive streak, it is called what? Primitive streak. First streak it is. So it is called what? Primitive streak. Okay. The primitive streak, where the primitive streak is formed, at that place only one shallow groove is formed. You know the groove? I show the picture in this video only. That is called groove. For example, if it is a straight, okay, if there is a streak, this is the streak. Groove means what? Groove means this. Groove means a shallow portion. Whenever the groove is formed, there is a folds. These are folds. It is groove. Okay, so like that, one primitive strike is formed on the bilaminar embryonic disc, longitudinal axis. Along with that, there is a groove is also formed. That means it's like a canal, how the canal look like near to the road. Okay, road is in a flat manner. Near either side of the road, maybe or one side of the road, there is a groove. That groove is called canal. In our homes also, you can observe this type of grooves in the washrooms, maybe at near the kitchen also, near the sink also, you can observe. And in our gardens also, we prepare grooves from one plant to another plant to supply the water. Like that groove is formed. Okay, primitive strike is formed, and at the same place primitive groove is formed. Whenever the strike is formed, that is the indication of gastrulation. Because up to now, up to the streak, the cells are confused, they don't have any goal, any aim to move. Whenever there is a streak, then they prepare a goal and they have a goal to move through that groove or towards the groove. Okay, so the primitive streak is the perfect indication of the gastrulation. Okay, next, the anterior part, in the anterior part it is the primitive groove. Okay, in the anterior part the groove is like this. In anterior part of the primitive groove there is a pit. There is what? A pit. You know already what is the meaning of the pit. Pit is a shallow portion, okay, like a funnel shaped. A pit is there. Where there is a pit, at this position, anterior position, the fold becomes more dense. The folds becomes more dense and more thickened. The thickened cells at the anterior end is called primitive node. You know nodes, in botany also you may observe the nodes in leguminaceae plants. The nodes are you can observe the nodes, Ranvier nodes in multipolar neuron structure also, internodes. So node means some bulging process. As like that here also one node you can observe, that is called primitive node. The streak is called primitive streak. The groove is called primitive groove. The pit is called primitive pit. All these are formed for the first time. So all these are called primitive. The first streak that leads to the first groove. At anterior end there is a first pit, primitive pit, and at that region only that thickens primitive node. Okay, clear up to here? Now what will happen? What is this layer name? This layer name is epiblast. On epiblast only it is happens. Okay, and underlying this hypo means underlying. Epiblast beneath the hypoblast. In epiblast there is a primitive groove. In epiblast, okay, I will show you this is the epiblast. In epiblast there is a primitive groove, primitive pit, primitive node, primitive groove, okay, and hypoblast. Clear? Epiblast is actually has all the stem cells. Epiblast have all the stem cells. They have to be differentiated now. They have to be differentiated now, and they have to move to their particular places. Up to here there is no differentiation. Okay, these epiblast cells start migrating. They converge. Converge means what? They communicate with one another and all come together in one particular place. All are together to go to one particular place. This is called converge. All the cells which want to go, all the cells come at one place. That is primitive fold. They all come to this place. So I'm going to show this in this manner. Okay, this is the epiblast. Epiblast cells are somewhat columnar cells. So because of the groove, it comes like this. It is the groove. Okay. Beneath this, there is what? Hypoblast. In epiblast, there is a primitive groove. Okay. I am showing the yellow color cells. Some cells converge to the primitive folds and they involute, they enter, okay, they descend, they involute, they descend into this primitive groove and they come down. They come down and they replace this purple color means hypoblast. Okay, this purple color hypoblast is replaced by this yellow colored future endodermal cells. Now hypoblast is replaced by the endodermal cells which are originate from the epiblast. So epiblast sends some cells to the primitive groove that is going to replace the hypoblast. Now it is not hypoblast, it is the endoderm. Remember, endoderm is the first layer which is going to form in this embryonic development. A perfect endoderm is now formed. Still ectoderm, mesoderm is not yet formed. Okay, once endoderm is completed. Next, another special cells, red color I am giving. Another special cells also same. They converge at the primitive folds. They involute or convolute into this primitive groove, and they take place between the endoderm and epiblast. And now it is called mesoderm. It is called what? Mesoderm. Okay, technically not yet the ectoderm is formed, but epiblast, the remaining epiblast remains as the ectoderm. What will be remain in the epiblast? The cells are considered as the ectoderm. So it is ectoderm. The blue color which is from the beginning, that epiblast only remains as a ectoderm. So now how many layers? Ectoderm, mesoderm and endoderm. 3 germinal layers is there. So this bilaminar is converted into trilaminar embryonic disc. Okay, by the process called gastrulation. Gastrulation means this cell movement. The cell movement is called, the dynamic process is called, okay, migration of the cells is called gastrulation. Okay, by the process of gastrulation, bilaminar embryonic disc is converted into the trilaminar embryonic disc. It contains 3 germinal layers: ectoderm, mesoderm and endoderm. Okay, it is not yet completed. Before it is converted into the ectoderm, after migration of mesoderm, within this there is a specialized white color cells. Of course they are not in white color, but I am giving for a clarity. There is some white color cells are there. They are called cardomesodermal cells. Cardomesodermal cells. Where you hear this word cardomesodermal cells? In first year structural organization, animal diversity 2 car data. In that time, in animal diversity 2 car data, notocard, in that cardomesodermal cells forms the notocard. Like that, the point is there that cardomesodermal cells are also present in the epiblast. Okay, this cardomesodermal cells come towards the pit. Up to here we discussed the primitive groove only. Now they are migrated towards the primitive node. Primitive node means the thickness of pit. Okay, and they converge there. Okay, and they involute. Okay, and through this primitive pit they come down. That means already here it is the epiblast. It is what? Epiblast. Already on the downside there is a endoderm is there. Here it is what? Endoderm. I'm showing the side view. Okay, I'm showing that side view. And in between the endoderm and epiblast, because of this gastrulation, mesoderm is also forms. It is the mesoderm. Okay, like these 3 layers are there from the side point of view. Now what happens? All these cells, the cardomesodermal cells, come down, okay, into the mesoderm, and they run forward. And they run what? They run forward like this. And it's going to become a notocard. They become very strong, and it is called what? Notocard. This notocard is the characteristic feature of the chordates. And in vertebrates, that means in our body, notocard is replaced by the Notochord is replaced by the vertebra. Vertebra is mesodermal origin, skeletal system that is. Okay. Here also notochord, it is also from the chordomesodermal cells. Okay. And of course the entire epiblast don't have this groove, don't have this pit, up to here only. So it is replaced by what? Endoderm. Between these two, mesoderm. And now here into the anterior side, from posterior to the anterior, here it is what? Notochord. Okay. So still some space is present here, still some place. Okay, that place don't have any groove, any streak, any pit. That means above the notochord there is no streak, there is no pit or nothing. Only epiblast is there. Only epiblast is there. That epiblast, after sending the chordomesodermal cells, it is converted into what? Ectoderm. Now it is ectoderm. So just imagine there is endoderm. On the endoderm there is a mesoderm. Between the mesoderm there is a notochord like this. Above the notochord, above the mesoderm there is a ectoderm. Okay. In ectoderm, here only the groove is there. Here nothing. It is an ectoderm. That means the plain ectoderm is present above the notochord. Okay. When notochord is formed, it stimulates the ectoderm. Yes, I am formed. Okay. I am actually going to provide protection for the neuron. So I have to develop the nervous system. Like that, it induces the ectoderm to form the nervous tissue. It induces the ectoderm to form what? Nervous tissue. Okay. So when it is going to stimulate that, here if you observe, this is what? Primitive groove. Okay. This is primitive streak and primitive pit. And towards this is what? A primitive node only. Above this there is a space. Okay. This space is actually ectoderm, a small ectoderm. Okay. And from here notochord is formed like this, and it stimulates, it induces the upper ectoderm to form a neural tissue. Immediately, because of the induction from this, actually it is called induction. Induction means what? Stimulation or inducing one type of tissue to form the other type of tissue is called what? Induction. That means what happens here? Mesodermally originated notochord stimulate ectoderm to form the neuron, to form the nervous tissue. So this is called induction. Best example for induction. A stimulation of one tissue by the another type of tissue is called induction. That is happened here. So by receiving the stimulation, what happens? I am showing here. Okay. It is the ectoderm. It is called what? It is what? It is ectoderm. The blue color layer is what? Ectoderm. Here only the groove is there, a primitive streak is there, and pit is there, everything. Here the notochord, which is actually stimulating the upper ectoderm to form the neural plate. So by receiving that signal, it forms one neural plate. It forms what? The neural plate. Plate will be formed first. That means the cells becomes ready and they become stiff and they are ready to form the neural tube. Okay, that is happened. After that, in this also one groove is formed. That groove is called neural groove, because neural plate, right? So in that groove is neural groove. When in neural plate one groove is formed, automatically what happened? Like this type of folds are happen. Folds are formed. These folds are called neural folds. And this groove becomes more deep. If it becomes more deep like this, these two folds come closer like this. If it becomes more deep, these folds come more closer and they attach with one another and they form finally one tube. Like this, neural tube is formed. This is called what? Neural tube. Actually, it is going to develop into the spinal cord and the brain. Neural tube. Formation of neural tube is called neurulation. It is not new relation, it is neurulation. But there is a new relation between the notochord and the neural tube. Notochord is going to give protection to the neural tube in the form of vertebral column. Okay, so this is neurulation. Few important terminology I am going to revise once. That is bilaminar embryonic disc. Only epiblast and hypoblast. At that time also, hypoblast covers, very important, hypoblast lines the yolk sac. It lines what? Yolk sac. At the same time, epiblast is going to cover the amniotic cavity. Amniotic cavity. Okay, and extra embryonic mesoderm is there. Before formation of the real mesoderm, extra embryonic mesoderm is also there, which is going to protect that epiblast and hypoblast. That is also covers this. Okay, why I am saying all this is it is very important to note down that embryo with extra embryonic membranes, like yolk sac, amniotic cavity, after later form the chorion and everything also. Okay, embryo with extra embryonic membranes is called conceptus. Conceptus means the production of the concepts. The production of the conception. Conception means fertilization, both fusion of male and female gametes. Okay, that is called conceptus, conception. Okay, and that leads to the pregnancy. And the embryo, resulted embryo with extra embryonic membranes is called conceptus. And this is called bilaminar embryonic disc. In this bilaminar embryonic disc, cells started moving after the formation of primitive strike, primitive node and primitive groove and everything. The movement of cells is called gastrulation. That gastrulation results in the trilaminar embryonic disc. Once the trilaminar embryonic disc is formed, the notochord, which is formed from the cardomesodermal cells that stimulate epiblast to become a neural plate. This is called induction. Okay, and because of this induction, the neural plate becomes formed, and the neural plate forms the groove also. Because of the groove becomes more deeper. Okay, the neural tube is formed. The formation of neural tube is called neurulation. Okay, so this is about gastrulation and neurulation. In next class, we will discuss how the extra embryonic membranes are formed, how the real true coelom is formed instead of this blastocele, and everything, the embryonic development, paturation, lactation, everything. Okay, I hope you understand. Thank you.